alignments, as commonly done, oversimplifies the evolutionary information which is inherent in a homology detection process. For the first two speakers on phyloseminar.org, I decided to invite two researchers who have thought a lot about the relationship between phylogeny and alignment. Marc Suchard, who has a Bayesian perspective, and Ward Wheeler, who represents the perspective of dynamic homology. So Marc has established many important results and methods in Bayesian phylogenetics. He is co-author of the BallyPhy alignment and phylogeny co-estimation package along with Benjamin Redlings. So more recently, he has also investigated the impact of alignment on phylogenetic inference with Wang and Hilsenbeck. He also has substantial interest in the inference of population history using phylogenetics, as well as adapting phylogenetic methods to study viral evolution. All right, take it away, Marc.
Well, thank you very much, Eric. Until the mid-'80s, most evolutionary biologists thought that all of life on Earth could be divided into two different groups, the bacteria and the eukaryotes, where the eukaryotes looked a lot like bacteria, however, they had intercellular organisms. Now, this sort of two-domain paradigm of life was turned on its head in the late 80s, very early 90s, by Carl Woese. And Carl Woese had the great idea of saying, let's try to reconstruct evolutionary history of all organisms that we know across the tree of life using shared molecular sequences. And Carl himself was most interested in ribosomes and looking at the 16S ribosomal subunit or its eukaryotic homologue. What Carl quickly discovered is that it appeared that life did not come from two different domains, but actually came from three. And the third domain has since been named, actually had this name beforehand, but it has since been adopted, the archaea. Now, this immediately sort of drove a number of contentious issues in genomics. And one of the first to sort of question the three-domain of life hypothesis was a long-term colleague of mine, Jim Lake, and a collaborator of his, Maria Rivera, here at UCLA. And Jim first looked at the archaea and thought something was a little amiss. You know, he noticed that the archaea shared properties with both the eukaryotes, those include us, and the common bacteria. In terms of their commonality with common bacteria, both the archaea and the bacteria sort of lacked organelles. However, when you looked at the structure of the proteins in which the genetic material was wrapped up, some archaea appeared to look more like eukaryotes than the bacteria themselves. So Jim hypothesized a four-domain of life scenario, which can be shown by sort of the two trees that I have on this first figure. And Jim's hypothesis is sometimes called sort of the archaeal hypothesis, or the eocyte hypothesis, which says that the archaea can be broken up into two groups. One, the eocytes, and the other group I'll just call the non-eocytes. And the eocytes themselves happen to be more closely related to the eukaryotes than the non-eocytes. This is in juxtaposition to the sort of three-domain of life hypothesis, which has the eocytes and non-eocytes as nearest neighbors to each other in a phylogenetic tree, as sort of shown from the archaeal tree that I list here. Now, this immediately caught the attention of such, reputable scientific venues as the Weekly World News, who published a paper that said humans are most closely related to slime molds, because they kind of like this idea that obviously humans represent eukaryotes, and our closest ancestor on the whole tree of life would be some slime molds that come out of the eocytes. Now, what evidence did Jim use in proposing this four-domain of life? Well, the evidence that he brought to light was, in many cases, anecdotal, or at least at that point lacked methodology to assign strong probabilities to the conclusions that he was trying to draw. He was looking at the EF1 alpha, or 2 protein. This is an elongation factor. It has different names in the bacteria and the eukaryotes. And it appeared that there was some evidence that the eocytes and the eukaryotes shared an insertion of a few extra amino acids. But it was very hard to tell that if this evidence would stand up to any statistical rigor. And one of the reasons it's quite hard to tell is we're looking at proteins that span the whole tree of life. So these are proteins that are very distantly related, and so they're hard to align. So in looking at a single alignment of those proteins, alignment might be extraordinarily biased by the methods you use to develop it. Additionally, at least at the time, there were no joint models that were able to combine both information from shared substitutions in molecular sequences and shared insertion-deletion events. So I'm going to start off by talking a little bit about the data that we're going to return to in this problem, and that's talking about the elongation factor. It's called EF1 alpha in the eukaryotes and in the archaea, and it's called EF2 in the bacteria. And as I mentioned, it's an extraordinarily conserved elongation factor. It's found across the whole tree of life, and it helps in the loading of tRNAs onto the onto the ribosome during the translational process. So it's a fundamental protein that's been around for a very long time. If we take a look at sequences of EF1 alpha or 2 from a number of different species, we'll notice that it ranges in length from about 400 to 460 amino acids. So immediately that suggests that over the space of the tree of life, there's at least been an increase or a decrease, we don't know how many times or when, of amino acids within this protein. And if you look at sort of similarity itself, any two sequences on average are about 26% similar. So that's sort of in the gray borderline region of similarity where we'd start to expect there to be problems given sort of standard methods for reconstruction. Some of the species that we're going to look at are sort of, perhaps not archetypal eukaryote. These are, us, sort of Homo sapiens. Eubacteria that you're probably familiar with, the E. coli that live in your gut. There are many others. It's the archaea that take a little bit of familiarity, and some that we'll examine are sort of those that come from very diverse or what we would think of traditionally as very inhospitable regions on Earth. For example, there are the sort of hyperthermophiles that live right on ocean sea vents at extraordinarily high pressure and temperature. In addition, we'll look at those that live in volcanic springs, and even sort of archaea, particularly the halobacteria, the halophiles, that live in extraordinarily high salt content, and those are sort of quintessential eocytes. You'll find them in like the Great Salt Lake, where the osmolality is large enough where even humans getting in there feel very uncomfortable within a minute or two. So I'm going to start by talking about sort of a statistical perspective to traditional phylogenetic reconstruction. And then I'm going to expand this view on phylogenetic reconstruction to include some models over these insertion-deletion events and see if we can actually start to build a model that derives from the observed data and not from some intermediate processes. So let's first begin with an intuitive example of how phylogenetic reconstruction works. This intuitive example that I have here involves 4 very common taxa that we're familiar with. And from each of these different taxa, I've listed some sequence. Each sequence from these different taxa consists of 10 different nucleotides. And what I've presented is a standard sort of 2-dimensional representation of what an alignment between these 4 taxa across these 10 sites might look like. And from an intuitive perspective, we might suspect that in the unknown tree that relates these sequences, that the human and chimp would be most similar to each other, since they differ at only one site, that sort of labeled in red here. There's an A to T transition. Actually, that's a transversion occurring. Whereas in this tree, we might think that the fly is the most distantly related from the other 3, given that it differs in several different locations, namely those in green. And so the branch sort of leading from the fly to the rest of the taxa, we might suspect would be a lot longer than the other things. In terms of this intuitive reconstruction, I've been talking mostly about looking at substitutions. So these are single residues, either nucleotides or amino acids or whatever sort of coding, whatever kind of coding state you've chosen for these, how these residues change from one to another. For example, somewhere between the human and chimp, there appeared to be a change from an A to T. But you'll also notice there's some extra sort of dashes that I've included in this alignment in the mouse sequence. Those are generally termed as gaps, and they're not really observables. They're simply placeholders that I've put in there to identify the fact that at some point in the evolutionary history, some residues have either been gained or lost. For example, we might hypothesize that these gaps arise from a loss of these characters sometime along the branch leading to the mouse from the rest of the species. Or, since this is an unrooted phylogeny, we might also hypothesize that somewhere along sort of the mouse branch, some nucleotides arose. So it could be either an insertion or a deletion. But let's move past sort of this intuitive understanding of how reconstruction works to a more formal statistical model. So a standard statistical model sort of runs as the following. Let's take each of these sites, for example, the 4 T's that I've boxed right here, and let's first assume that what we've made in the statement is a homology statement. We've assumed that sometime back in the past, these 4 characters shared a single common ancestor character, probably a T, and that T has been duplicated over time and passed down to the sequences that we've observed. So for each of these homologous sites, remember these are homology statements, these are guesses that we're making, we're going to assume that each of these sites are independent and identically distributed. Well, now what we have is for each site, we have basically a realization from a multinomial. If we're dealing with 4 taxa, and there are 4 possible outcomes for each of the characters, then there are 64 possible different patterns that we might see here. So we're going to take each of these patterns and assume that they're multinomially distributed by some unknown probability, where the probability is the probability of observing that specific type of pattern, given a generative process that says somewhere back in time there was a single character that has been bifurcated and slowly mutated over time. So this unknown probability is going to be a deterministic function of an unknown tree, branch lengths on this tree that are going to reflect sort of the expected number of substitutions along each of these branches, and a very simple continuous time Markov chain, which is a stochastic process that occurs conditionally independently along each of these branches that allows the character that exists for the state back in the history to change. And the continuous time Markov chain we can parameterize in terms of a rate matrix, and what we're really going to need for each of these branches are these finite time transition probabilities, or the probability that, say, nucleotide X here at this point mutated to the observed T at this point in time T. And that's a simple integration of the process, and it's an entry in this matrix exponential. Given all those matrix exponentials, it's a relatively straightforward process to calculate the multinomial probability. So let's start with a given tree, and let's assume and start with an observed pattern at the tips, for example, A, A, G, and T. And let's assume for the moment that we know what the state of all those internal nodes are, or what the character at that site, how it existed back when 2 of the branches merged together. Then we can write down the multinomial probability of the observed pattern as simply the product of all of the transitions along each of the branches, summed out over the marginal probability of one of the states. For example, we have the probability of some state Y mutating to A in, where did that one go? In time 4. Likewise, we have Y mutating to A in time 1. Equivalently, we have the transitions from X to G and X to T and X to Y. What we don't know, however, are what are the unobserved values of X and Y, and so we're going to integrate, or in this case it's a discrete sum, over all possible realizations. And that sort of forms the basis of the phylogenetic likelihood, or the unknown or the deterministic probability that runs into the statistical multinomial model. For any n sort of species, there are a sort of an exponential number of trees. For the 4 species I've listed here, the number of sort of unique trees is 3, but that number grows very likely. So one can sort of pick their own favorite procedure, given a statistical likelihood, to select among the different possible trees, if we were interested in reconstructing what the most likely tree is, or a probability distribution over those trees. And you can do that via Bayes' theorem. We've written down the conditional sampling density of the data given the unknown tree and the parameters. You can invert that to get sort of probability distributions that are of interest to your problem at hand. But there's a fundamental problem with the type of inference that I just described, and that runs in the following. In a sense, we're defining a statistical model over a multiple sequence alignment. However, the multiple sequence alignment itself is not an observed quantity. You do not go out in nature and observe sequence alignments. As a matter of fact, sequence alignments themselves, or at least multiple sequence alignments, aren't even real entities. They're not even a parameter of the underlying generative process that gave rise to the sequences that we observe. Multiple sequence alignments themselves are simply low rank or low dimensional approximations of a huge number of homology statements that we're making about the observed characters. What we do observe in nature, however, are the sequences themselves. So the directly observable are the sequences, not the alignment. Traditional, or at least most traditional, phylogenetic reconstruction methods work in a sequential approach. They start with the direct observables and then use standard alignment software, and here I've just listed 2, +ALW or tcopy, to arrive at a multiple sequence alignment. These approaches then generally condition on that multiple sequence alignment being true. Now that's a rather spurious conditioning, because you're conditioning on something being true where that something doesn't really even exist in nature. So if we condition on the alignment being true, then we've developed a huge number of methods and spend a huge amount of computational time deriving a probability distribution or point estimates of the unknown tree given that inferred property. The problem, in many cases, with this sequential approach is this first step that starts from the observed data and gives you a multiple sequence alignment generally involve very simple models of evolution. So one might spend a considerable amount of time improving our current models of evolution that we use in going from the alignment to the tree, but in the end, the answer that you're going to get could be very highly biased by the very simple evolutionary models that went into this initial step in the sequential estimation approach. And that is particularly true for alignments that are very different. Well, it's actually very true on two different scales of evolution. For very distantly related sequences, they are prone to alignment bias. And this has been known for quite a long time. And the types of problems that we're going to talk about mostly in this talk come from that example. At the end, I'm going to talk a little bit about sequences on the other side of the evolutionary scale, those that have been sampled over the period of sort of months or weeks of rapidly evolving pathogens, in which case there are other issues of alignment bias that come in. But the real trouble is these initial steps that start from the direct observables generally assume some very simple guide tree and naive evolutionary models. And the answer that you get at the end could be very biased by those initial assumptions. Well, this is kind of like a chicken and the egg problem, which came first? And the solution to the chicken and the egg problem is that neither came first. They both arose at the same time. So instead of taking a sequential approach to estimating the underlying tree, if that's what we were interested in, one can consider inferring both the homology statements, or the low dimensional projection of those statements, what we call a multiple sequence alignment, and the phylogeny at the same time. And this is by far not a novel idea. People have been talking about trying to do this since the early 90s. There have been some very successful approaches at this type of simultaneous inference since sort of the late 90s on through. Many that you've probably heard of, and you'll hear Ward talk about considerably on the next talk, have to do with sort of optimization alignment, or POI. Additionally, there has been considerable work in developing stochastic models that give rise to multiple sequence alignments given sequences. So how the insertion deletion process occurs along a tree. Most notably among these come from Jeff Thorne and his early work in the 1990s, so the TKF 91 and 92 models, and their use in inference of evolutionary trees. However, these more rigid sort of birth-death developed processes come with sort of substantial computational burdens, and there's also some difficulties in terms of sort of some forbidden positional homologies that we are pretty sure exist in real life but are not allowed under these simple birth-death processes. So instead of thinking about an alignment as an observable or as data, let's conceptualize it as a random variable. Now, I don't want you to get the idea that it's a random variable that actually exists out in nature. It's not. It's just a low-dimensional projection of a whole bunch of homology statements. But nonetheless, it is the sufficient statistic that we need in order to go from the sequence to the tree itself. So here's what an alignment might look like as a random variable. Let's assume that our observed data are four different sequences, where the first three observed sequences are of length 4, and the last sequence is of length 3. Now let's assume for a moment that we know the relationship amongst these 4 sequences, and that's given by the tree here, where 1 and 2 are nearest neighbors, 2, 3, and 4. The alignment itself is actually a projection of these sequences into a 2-dimensional array. So here's an example of such a projection, where each entry in the projection either says pull character 1 from sequence 1 and lay it down here, or pull character 2, or don't do anything, pull character 3, and pull character 4. So we can project our sequences onto the series of homology statements. Notice this is making homology statements. This is saying that character 1 in sequence 1 is homologous to character 2 in sequence 2. And we get the observed data itself. Now, let's also introduce the missing data in this problem. And for the phylogenetic likelihood itself, the missing data are the sequences at each of the internal nodes. Let's assume for a moment that we know that the missing sequence at Y5 is actually of length 3, and the missing sequence of Y6 is of length 4. Then we can project the homology of these unobserved sequences to the observed tips as well. So here is an example of just one possible homology. So T, C, and C are all homologous to each other, and they're also homologous to 2 unobserved sequences at the internal nodes. Now, this data augmentation procedure takes the standard alignment, which is a low-dimensional projection of the homology statements, and augments it into the larger space, which includes all of the homology statements that are occurring along this tree. For example, by doing this augmentation, we can actually pinpoint where on the tree each of the insertion and deletion events occurred. For example, this green bit right here specifies that there is a character that exists at Y5 that is homologous with the 2 C's that we observe at the end of the tree. However, there is no character at Y6. So somewhere in this history between Y5 and Y6, there was an insertion deletion event. So one of the advantages of this type of data augmentation is we're now able to pinpoint on the tree where the insertion and deletion events occurred. Now that's a major improvement over most progressive alignment procedures. Most progressive alignment procedures consider only this lower dimensional projection. And in doing so, they intrinsically assume that gaps that exist in this alignment, they're scored as if they had occurred on the external branches of a tree. Or another way to think about that is none of the insertion deletion events could be shared by descent. However, by augmenting the space with the sequences at the internal nodes, we're able to say that these two characters are homologous by descent, and we can use that information to improve our resolution that C and C should be nearest neighbors to each other on the tree. Because there was a single event here that then gave rise to a single character, perhaps a C, that was shared by descent. Whereas if we were using a progressive alignment procedure, it would say that there were two independent insertion deletion events along these branches, and that doesn't add any information to putting the C's together. So in doing this sort of data augmentation, not only do we get around the fact that insertion deletions can be shared by descent, but we can begin to envision using that information of indels that are shared by descent to improve our phylogenetic resolution itself. Also, by augmenting the state space such that we have the number of characters at each of the internal nodes and their homology statements, we've in a sense separated the data sampling density of the characters from the insertion deletion process itself. And that's because if you were to look at the peeling algorithm that we had before, in doing the integration over an unobserved character, that integration is the same as there have been, it yields the same number as if there were no character at that sense. So we've taken a very complicated sampling density over alignments and phylogenies and broken it down into the traditional Feldenstein likelihood on a tree for the observed sequences and the internal node states, and a conditionally independent process of how these insertion deletion events occur on each branch. So we have our standard substitution likelihood, which over the last, well, since 71, over the last 30 years or more, I've spent a lot of time understanding, times in a sense what a Bayesian would call just a prior over the gaps. But we want that prior to have some underlying generative process that we can understand and parameterize it in terms of quantities that we'd like to learn about from the data. So what might a very simple model for a gap prior look like? And the simplest model that I can think of works in the following way. We're going to, and I need to keep my mouse from doing that. Let's decompose each of the homology statements that we had in that matrix on the other slide into a number of pairwise alignments on each branch of the tree. So we can take the augmented multiple sequence alignment and rewrite it as a number of pairwise alignments on each branch. Then we can come down with a very simple process that gives rise to the pairwise alignment of sequences. And one of the simplest processes that you can think of is defining these pairwise alignments as arising from a paired hidden Markov model. So a paired hidden Markov model omits a number of states, and it's paired because it omits observations on both ends of the branch, and it follows a very simple process. So here's one of the simplest hidden Markov models that you might envision for such a process. You can obviously come up with much more complicated time-dependent ones. So this hidden Markov model basically omits 3 different states. It omits for each set of characters a match state, that says that these two characters that I'm admitting on either side of the branch are homologous to each other, or it emits into insertion deletion events, which says that there's a character on one side of the branch and it's not homologous to anything on the other side of the branch. And of course there are two because there are two ends of the branches and it could be either way. We could parameterize this hidden Markov model in, well, in fewer than 2 parameters, but here's sort of a 2-parameter parameterization, where there's a probability of an indel occurring. So you're in a match state, and you then, the next step emits in one of these indel events. And then there's a probability of extending an indel. Using this 2-parameter instead of 1-parameter approach, you can quickly incorporate some very nice aspects, for example, affine gap penalties. One might expect that insertion deletion events that are not independent for each character, but that the probability of having a 2-character indel event is not the probability squared of having a single indel event, but it's just slightly larger, or it's dependent on the it's at least not as large. One can expand the number of admission states here, for example, these in series with different sort of epsilon values, to get a nice, more realistic distribution on the gap lengths that you might expect. For example, in coding regions, you might expect that insertion deletion events of length multiples of 3 are much more likely than not. Then you can add on three of these emissions in series and come up with a nice probability distribution that puts a much greater mass on 0, 3, 6, 9 than it does on 1, 2, etc. Also, we can start to introduce sort of time dependence into this process right here. Here I've said there's some probability between each of the events, but we might want to make that a function of what the branch length is itself. And we can return to that in discussion later, but it's not really necessary for understanding the whole model. Well, believe it or not, that actually specifies a whole probability distribution over the space of alignments and phylogeny, just given the observed sequences themselves. I'm going to take a Bayesian approach and say I'm interested in estimating the joint distribution of alignments and homology statements given just the observed sequences. And one of the reasons I'm going to do that is so that I can get sort of natural measures of uncertainty on these very discrete, very complicated objects, the topology or the evolutionary relationships amongst the sequences, and the homology statements themselves. One thing I sort of breezed over very briefly before was this one used to think of the space of phylogenetic trees as very large. Well, actually, it's puny compared to the space of all multiple sequence alignments. Previously on a slide, I had 4 sequences where each sequence was of length 3 or 4. And just under that trivial example themselves, there are over 1 billion possible multiple sequence alignments. So as you start moving into normal-sized problems, hundreds, thousands of nucleotides, tens, hundreds of taxa or of sequences themselves, the number of possible alignments is huge. And so estimating a single point estimate might not give you a very good description of the data itself. Additionally, one nice aspect about a Bayesian perspective here is we can very efficiently marginalize or integrate out the parameters that we're not particularly interested in. For example, you might work in the comparative genomics field where you have a very good idea of what the alignment of what the topological relationship between, say, humans, mice, and rats is, but you might be interested in aligning sequences. So in a sense, you just want the marginal distribution over the alignments given the data and some information. You can easily integrate that into with very little change to the algorithm that you have in terms of drawing your inference. Alternatively, you might actually not be interested in what the homology statements are, but you just want an unbiased estimate of the topology. So you can very effectively marginalize out the alignment. And I think that that's a bit of a misconception right now in the literature about what simultaneous inference of alignment in phylogeny mean. It doesn't just mean you get one. It gives you a probability model from which you can draw inference for a marginalization of one or the other, where you're integrating out the bias that might be introduced by conditioning on one, or the joint distribution, depending on the problem at hand. To do this integration, I'm going to use Markov chain Monte Carlo. Markov chain Monte Carlo has been very well developed for sampling the evolutionary tree, given your substitution process. So this was the continuous time Markov chain that described how sequences mutated amongst themselves. And the alignment. Those spread back to a number of sort of independent developments in the 90s. And there's also been a fair amount of work on how to sample an alignment or how to sample these homology statements given a tree. And those stem back to some very wonderful work by Ian Holmes. So what Ian's key insight to the problem of, I know the tree, how do I sample the alignments, comes down to breaking this whole tree up into a number of very small updates in a Gibbs cycle. So instead of trying to infer the homology statement amongst all of the sequences from the ends of the, I'm trying to get this in the camera, the ends of the tree at the same time, you can just look at a small portion of the tree and do small updates. And that changes the amount of computational effort that you need to make in each of the updates. For example, you might just pick one of the internal nodes and resample what its homology is to each of its neighbors. Or you might pick a larger neighborhood with which to do this resampling. So obviously there's quite a bit to be gained in terms of decreasing autocorrelation in your Markov sampling if you take a big jump, but the computational effort is very high. So somewhere in the middle ground is going to be some optimal way of sampling the alignment given the tree. And then we can just iterate back and forth in some ways to hope to converge to the joint distribution of both. So if one has a very large tree, how do you decide how much of the information in terms of the homology statements you want to update at one point? And this really comes down to deciding how many of the internal nodes do you want to resample in terms of their existence of sequence characters or not. Well, the way that one does that sampling is by using a forward-backward algorithm. It's generally known as dynamic programming in the computational biology literature. Mathematicians like to call it the sum product algorithm, but it was really the existentialists who kind of understood forward-backward the best. It was Kierkegaard who said that, you know, life is lived forward, but only understood backward. So we're going to calculate our likelihoods forward, and then, you know, basically draw a sample from it coming back as a realization. And these allow us to take exponential searches over all possible alignments and reduce it down to a polynomial time algorithm. But the larger the space that we're trying to integrate or sample over, the greater that polynomial order. So what Ian originally did was to say, I have a huge tree. So this is a huge tree I'm looking at, and this node right here in the middle is just one internal node in that tree, and these are all the subtrees that tangent to it. And Ian said, I'm just going to resample the character. Is it a character or not? And is it homologous to sequence 1, 2, or 3 or not at each of these internal nodes in sequence? And that leads to a very low order or a very fast polynomial time algorithm. Sort of it's just linear in the length of the sequence. So it's very fast to do. But unfortunately, you don't move around the space very quickly. And as a matter of fact, it leads to rather poor mixing. And here is an example of why it's hard to move through that space. Let's say our current state has two characters that are unaligned to each other. Notice this is not homologous because it's in the first position to this character that's homologous to the second position. If I'm only resampling homology statements at this internal node, in order to get to the aligned state, I need to go through a very unfavorable intermediate. So here are two indel events, and I want to get to one indel event. In order to do it, I need to go through a 3 indel event state. So it's going to be very slow computationally to use that approach. Now, Jotun and his group very quickly pointed that out and proposed a cubic algorithm. And in that cubic algorithm, we actually sample the homology statements of the first of the three different subtree nodes around Y4 at the same time. And that allows you to very quickly go from this state to this state. However, its computational order is very large. Well, as I like to tell most of my students, if you're going to do something and it's linear, definitely do it. If it's cubic, don't do it at all. If you can come up with something that's quadratic, think very highly about it and explore it further. Here's some wonderful insight that Ben Redling's had in this problem. If this is too hot and this is too cold, maybe there's something in the middle that's just right. And the idea behind this is instead of resampling the whole relationship amongst these 3 subtrees at the same time, we're going to leave the homology relationships amongst 2 of them fixed and just resample the relationship from the third to the first 2. Now, here's an example of the space in which we're trying to resample through. So I am representing here an alignment path along those 3 branches as the alignments along those patches as an alignment path. So an alignment path takes you through your sequence space, and basically as the links in the path give you each of them homology statements. Now, if we have 3 sequences that we're trying to align to each other simultaneously, we need to explore this whole cubic box of possibilities and assign a probability to each path that goes through this cubic box, and then draw one based on that probability distribution. Well, Ben's insight into the problem in terms of restricting 2 of them to be fixed to each other was basically saying, instead of sampling from this cubic space, leave the relationship amongst sequence 1 and sequence 2 fixed. So instead, we're just sampling along this wall in this space, or you're sampling amongst this 2-dimensional object. And that leads to a quadratic algorithm. Now, does the quadratic algorithm help? Well, here is an example. So here are trace plots of an MCMC sampler. So these are points that are proportional to the posterior probability, or this is, well, just the log probability of the joint state that we are at each step along our Markov chain as we're exploring this space. And here is the traditional way of sampling using an L1, using the linear and the cubic algorithm. The cubic algorithm is extraordinarily slow. And these are autocorrelation plots from 10 different runs. And you say, you know, we run it for a very long time. This was on the order of about a day. And we still don't even see convergence between the independent chains themselves. Whereas if we were to turn on this Goldilocks kernel, then you see something very striking. First, all the chains converge very quickly. And I really mean quickly. Look, the x-axis here are on a total different order of magnitude. This is 350 and this is 3500. So what do we find in terms of the MCMC transition kernel and its effectiveness? Well, the burn-in decreased dramatically, right? It went from some number that we couldn't estimate because we got bored to a couple minutes. And what I haven't shown here, but is also true, is you significantly decrease the autocorrelation amongst your samplers. So you don't have to run the chain quite as much to get as many approximately independent realizations from the process. And for one of the data sets that we're going to look at in a couple minutes, this led to about a 70-time runtime improvement. So it actually turned this problem into something that you can run in reasonable time. Yes, there's a question.
So Daniel, I think it's Dan Ford, would like to know, he says it's 70 times faster in terms of iterations, but how much more expensive is it in each iteration? So what sort of, what length of sequence would this break down for?
Yeah, so that was actually 70 times faster in terms of runtime and not iteration. So that takes into account both the fact that the algorithm runs a lot slower. For the problem that we're going to look at with that 70-fold number, those sequences are on the order of about 300 amino acids long. We're in a lot of work right now of other ways of speeding up these types of computation. Unfortunately, I don't have time to talk about all of that at the moment. But yes, it's no longer linear, but that was total runtime. So that's basically the product of the amount of time that each of the steps takes times movement. And we ran them so that we had the same number of sort of effectively independent samples in that sense. Now, there's a very big lie of omission that I went through in describing this sort of prior that we're putting over each of the branches, and that is they really aren't independent of each other. And the sampling that I've been proposing assumes that they're independent. They are conditional on the fact that the two alignments on either side of a branch, so here are my two branches, have to admit the same number of sequences at the internal node. So there's coupling between the lengths of the sequences that they emit on the internal nodes. So the real distribution is really these independent Markov priors normalized by a factor that involves sort of the restriction that all of them are emitting sequences that are of the same length. The problem with this normalizing constant is it actually depends on the data. It's not just a constant that doesn't change through the algorithm. So it's actually extraordinarily impractical to do a Gibbs sample, as if I kind of made the impression that we were doing Gibbs samples directly from this normalized distribution. However, and this is a good lesson for a lot of problems, is forget the normalization for a moment and just draw a Gibbs sample from the distribution that actually says that they are product-wise independent, conditional on giving the right sequence lengths. Well, this is a nice Markov, this is quite amenable to the Gibbs sampling that we're doing, and then use that Gibbs sample as a realization for a Metropolis Hastings step. So you basically draw a Gibbs sample from a distribution that's almost what you want, and then do an accept reject. And it turns out in this problem and in a large number of problems, those rejections are quite rare. So the only expense to the algorithm is going through and recomputing that normalizing constant, which in this case is quite pretty to do. So here is a very small example of the type of sort of uncertainty you might expect in some sequences that are very similar to each other. Here are a number of envelope sequences taken from some SIV virus. And at the top, what we have is the Clustall W alignment. And the Clustall W alignment seems to think that it actually puts all of the insertion deletion events on the tips. But if we were to be smart and reinterpret it, we might say that there's the 1, 2, 3, 4, 5 indels that it's trying to put in here. Well, here is an estimate of the most probable alignment under a sequential model, under a joint model, and it actually provides a more parsimonious description of the data with 1, 2, 3, 4 insertion deletion events. But you shouldn't take this as a good estimate of the true history, because there's a considerable amount of uncertainty around it. And here's some sort of poor animation to give you some, or not, to show you some random samples from the posterior. So this is old animation. And as you can see, there's quite a bit of uncertainty about where these insertion deletion events are occurring. And that's not surprising, because if you look very closely, which I don't expect you to do, this is a region of a number of repeats. But what is not very uncertain is the number of events, and the fact that these three sequences share one insertion deletion event by descent, and these two sequences share an insertion deletion event by descent. So we might want to come up with a way of summarizing some of this uncertainty in the alignment itself, or at least in our low-dimensional projection of the alignment. So here is an alignment uncertainty, or gold plot, where what we've done is taken an estimate of a very probable alignment and then gilded each of the characters in this alignment with a probability statement that says that this character aligns to something at the sort of the mythical root of the tree. So the way to interpret that is areas of high certainty that characters are homologous are in red, and areas of uncertainty are in colors sort of shading throughout the rainbow. So what you see is there's very high probability that this insertion deletion event exists. However, what is uncertain is, did it start here or does it start over here? And that's because there's a CAA repeat that could equally likely be over on the other side. The same thing is true over these other insertion deletion events. Where they occur is uncertain, but from a statistical perspective, if we're integrating overall possibilities, we just want to ask, is there evidence that there's an insertion deletion event shared by descent? Finally, you might recall that I said, well, we could iterate back and forth between sampling the alignment given the tree and the tree given the alignment. However, trees and alignments are highly correlated. And so that might lead to a very slow mixing Markov chain. One way to improve that mixing is to accept the fact that they're highly correlated and do what's called a collapsed Gibbs sampling approach. So we're going to generate from the joint distribution of the alignment in the tree by first integrating out a small part of all the alignment statements. So this is how you might do a nearest neighbor interchange. First, we integrate out over all possible alignments between these four subtrees. Then we change those subtrees amongst each other, and then resample an alignment given the change. So we've integrated out the correlation and then introduced the correlation back in, but correlation that's more consistent with the step that we just took, which allows you to move through these sort of very correlated spaces much easier. And there's similar procedures that you could use for like global changes on a tree, like SPR moves. So let's first start with an example where a reasonable answer would be, there's no information in the data. Don't try to give me really strong support for an answer. I know that's just bias. And one place where that might come from is trying to reconstruct the evolutionary history of a number of species based on a 5S RNA sequence. And a 5S RNA sequence is about 100 nucleotides. And most evolutionary biologists would say that they're relatively uninformative. So if you were to reconstruct a tree from these sequences, you should get no information about the tree. The question is, we should get no information, but if we use a sequential approach, maybe we get really strong information for the wrong answer. Here's an estimate of what the posterior distribution of alignments, or at least the alignment uncertainty, for a number of 5S RNAs across the whole tree of life might look like. And what you immediately see is there is some evidence of fairly strong statements of homology amongst the sequences on the sort of the 5' end of the sequence. But as you move to the 3' end of the sequence, there's no information in there to assign homology or draw inference about the evolutionary tree itself. But, what if we had ignored that and just gone through with the sequential estimation approach that is most common? We take our sequences, we throw them into ClustalW, for example. It's a very popular program, and then do Bayesian inference over the trees that we get. Well, here are the results that you find. And what I've listed here are posterior probabilities of different partitionings of the data. This is conditional on the Clustall alignment. And the first thing that you see is conditional on the Clustall alignment. There is supposedly very strong evidence in the data for E. coli, these are bacteria, being nearest neighbors to Homo sapiens. And no evolutionary biologist would say that that's the correct partitioning of the data. However, if you look at the data, if you look at the guide tree that Clustall first assumes, the first thing it does is it says those two are nearest neighbors, puts them together, and then no matter what sophisticated analysis you do given that alignment, you get that biased result back. However, if you do a simultaneous analysis where you integrate over all possible alignments, you find that absolutely the data provides no evidence for any partitionings, which is what we would expect. So in a sense, by integrating over possible alignments, we've decreased our possible bias that that type of conditioning could introduce. But you get a lot more out of a simultaneous inference than just decreasing bias. In most statistical problems where I take a statistical problem and I add in more variables, you generally decrease bias, but you do so at the expense of increasing variance or getting less resolved inference. Well, that's not necessarily the case here. We've expanded our model, but we've expanded our model in such a way where we're actually extracting more information out of the data. So not only can you decrease bias, but you can also get more certain inference by drawing information from insertions and deletions that are shared by descent. Here, we're finally going to return to the EF2 problem and address Jim's question of the eocyte tree. Well, the first thing I'm going to say is doing a joint analysis over the EF2, you find very strong posterior support for the eocyte tree that clusters Homo sapiens with the eocyte, which in this case is a Sophilopus. Where does that really strong support come from? Well, here is an alignment uncertainty plot of all of the data. And I know you can't read it. We're going to focus in on small bits of it on the next slide. But what I want you to appreciate is there are regions in the gene where we can make pretty strong homology statements. And from those regions, we can extract substitution information. However, there are also regions where there's quite a bit of uncertainty in the homology statements. And in those uncertain regions, we do not want to condition on one wrong alignment and get false inference. Additionally, there are some insertion deletion events that appear to be shared by descent, and perhaps that's where the information is coming from. So I'm going to blow up one of those regions right now that we can look at. So immediately, the joint inference approach identifies an insertion deletion event that's shared by descent between the eocytes and the eukaryotes. And that's these sort of 7 or 8 nucleotides right here. Now, those are the, amino acids. Now, those are the amino acids that Jim initially identified in 1990. But he didn't have any way of sort of quantifying the support that they added to the data. Yes, please. Oh, yes. So we've been able to incorporate that automatically into this point. Additionally, something that Jim didn't notice, and this irks him all the way to the day, because in his alignment it wasn't there. There's actually another shared insertion deletion event just a couple amino acids further that led support as well. Now, it's been quite a while since I've given this talk, so I'm not quite up on the right sort of speed that I'm going. So I'm going to say that the shared indel actually is what increases our support for these multiple radiations of the eocytes themselves. Matter of fact, there's a shared insertion deletion event that happened after Pyrococcus divided that changes our posterior support from we can't really draw much of a conclusion to a fairly strong conclusion, which happens to sort of weigh in favor of these multiple radiations. So, you know, what are some of the big take-home messages? Well, an important first take-home message is that an alignment is not a real object. It is, in many cases, a multiple sequence alignment that we see printed out is a low-dimensional projection of a whole bunch of homology statements. And it's actually on those homology statements that we want to be drawing inference. We don't want to optimize on a quantity that doesn't really exist. So what you can do is you can develop a joint model that allows you to write down a stochastic process over the homology statements and the substitution processes at the same time. This allows you to produce sufficient estimates of the phylogenetic tree, and you get the additional benefit of using information from shared insertion deletion events to help resolve parts of the tree that you weren't able to grab before. Additionally, by doing this integration, you can avoid bias that gets introduced by conditioning on what might be a false alignment. And it also enables you to put sort of realistic assessments of the uncertainty on the tree and on the homology statements themselves. In order to make this all work, one had to think rather carefully about how to develop transition kernels over this space. But we're moving to the point where you can start approaching larger and larger problems. And at least from a biological perspective, we're finally able to statistically incorporate the shared information of that indel that Jim sort of ad hocly pulled out of nowhere and became very contentious in the early 90s, and actually say it does provide good evidence for the eocyte hypothesis. It also starts to provide evidence for multiple radiations of the archaea. So we might not even exist in a in a four taxon exam, in a four domain world, but maybe a much higher domain world. No work is ever done in a vacuum, and almost none of this work, unfortunately, was done by me. Most of this methodology and ideas have been developed by a former graduate student of mine, Benjamin Redlings, in collaboration with some material that I didn't cover on rapidly evolving pathogens with Vladimir Minin. Of course, a good deal of thanks goes out to a long-term colleague of mine, Jim Lake, here at UCLA. And financial support is always helpful. I know it's starting to sound a little bit like an NPR commercial. We'll get something from, I guess, the Doris Day collection at some point, but your tax dollars are always helpful. And I'd like to answer a few questions now, if I can.